Just a brief introduction for our seminar guest. Dr. Maria Starling is a biologist who holds a PhD in sanitation, environment, and water resources. As an early career adjunct professor at the Department of Sanitary and Environmental Engineering at the Universidade Federal de Minas Gerais. My Spanish or Portuguese is not good. She's currently a junior Fulbright Visiting Professor at the University of Minnesota, the School of Public Health Division of Environmental Sciences. UFMG is a leading institution in Brazil with more than 50,000 students. Professor Starling is a member of the graduate program in Sanitation, Environment, and Water Resources, and her research has centered on pollution characterization, control, and prevention, mainly focusing on water quality monitoring and assessment, biomonitoring, ecotoxicology, as well as industrial and municipal wastewater treatment for removal of contaminants of emerging concern. So welcome to Dr. Starling, and thank you so much for joining us for our BBE seminar. I will let you take it away. And just to preface everyone, Dr. Starling is on a tight schedule as she is at a current conference for Fulbright scholars, so we may not be able to get to questions, but you are free to email her. I'll send a follow-up after this meeting with her information. Thank you.
Thank you. Thank you for having me here. So I'll go ahead and just remove these things out of my screen so that I can see my screen properly. So good evening, everyone. So as already introduced, I'm Professor Maria Clara Starling from the Federal University of Minas Gerais in Brazil. And I'm here during the fall semester as a junior visiting Fulbright scholar. So in my seminar today, I will talk a little bit about my background and then how I got to work with contaminants of emerging concern and in my academic path. Then I'll talk about contaminants of emerging concern themselves, the initial occurrence and fate worldwide and in Brazil. And then the application of solar advanced oxidation processes as post-treatment or advanced treatments for removal of contaminants of emerging concern from municipal wastewater treatment plant effluent, and especially removal of antimicrobial resistance. So antibiotic resistant bacteria and genes, which are spread nowadays from wastewater discharge, and then some final remarks. So as Rachel said, I'm a biologist. So my undergraduate was in biological sciences at the Federal University of Minas Gerais. So I graduated in 2013. And then I began my master's in the graduate program in Sanitation, Environmental Sciences. This is the abbreviation of Meio Ambiente in Portuguese, so MA, and Water Resources, Recursos Hídricos in Portuguese, so it's RH, during which I worked with solar advanced oxidation processes for the treatment of textile wastewater, aiming at the reuse of water in the industry. And then I just kept going for my PhD, also in the same graduate program, with a mobility period at the University of Lille in France through Erasmus in Brazil. And in the PhD, I actually worked with contaminants of emerging concern and their removal from water and wastewater. Right after I finished my PhD, there was a position opened at the Department of Environmental and Sanitary Engineering at the Federal University of Minas Gerais. So I applied for that, and I studied really hard for the process, and I was approved. So I became a professor in 2019. And now I'm here as a Fulbright Junior Visiting Scholar at the University of Minnesota. I got here in August. Unfortunately, I'll be going back home in a few weeks from now. But it was a great experience. So it was all worth it, and I would do it again and again. So you've seen that the Federal University of Minas Gerais is a big part of my academic path, and I'm going to talk about it for those of you who don't know. So we're here at the University of Minnesota. I'm actually not there now, but you're there. And this is Brazil. So the Federal University of Minas Gerais is in the state of Minas Gerais, which is located in the mountains. The capital is Belo Horizonte, which is this city, which has 2.37 million inhabitants. It is 1 hour from Rio by plane, 1 hour from São Paulo by plane. So the same region of both of the cities that most people know in Brazil, the southeast region. But whenever we talk about Belo Horizonte, it seems like no one knows where it is. Until I say, it's the soccer stadium where we lost 7-1 to Germany in the last World Cup, which was in Brazil. And then everyone can connect to the city somehow. And actually, this unfortunate soccer stadium is actually just by our campus. So the soccer stadium is here, and the Federal University of Minas Gerais is all this area circled in blue. It has been founded in September 7th, 1927, and it has a community equivalent to 72,000 people, including students and staff. There are 77 undergraduate courses, 80 graduate programs, and 900 research centers in our university. I'm based here, which is the engineering school. So this building, all of these trees that you're seeing here are mango trees. So in the summer, you have to be really careful where you park your car because you're going to have mangoes fall into your head. The engineering school was founded in 1911. It has 11 engineering courses and 60 research domains. And this is where the Department of Sanitary and Environmental Engineering is located. Actually, this is actually the window from my office. So I can see all the mango trees back there. It's really nice. And this department is actually a reference in sanitation and environmental sciences in Brazil and Latin America. And I believe one of the reasons why this occurs is because we have a sanitation training center inside the wastewater treatment plant in town. So it's the largest wastewater treatment plant in the city. It treats wastewater that comes from 1.5 million inhabitants, and PhD and master students, they actually have the chance to perform their projects using real wastewater that's coming to this plant in this pilot plant scale reactors. So this made us a reference in sanitation and environmental sciences in the country and in the continent. There are 21 professors, 11 women, so we're half there, and 9 laboratories. Along with the Department of Hydraulics and Water Resources, we form the graduate program in Sanitation, Environmental Sciences, and Water Resources, which is the graduate program through which I got my master's and my PhD, and I supervise master's students now. So this program has a maximum score in the Brazilian Research Evaluation system, which is 7 out of 7. This is important because the highest your score, more funding you get from the government to do research. And this is evaluated according to number of publications, quality of the publications, impact factor, and age factor of researchers, as well as the reach and internationalization of each graduate program. There are 10 research lines and 5 research groups. And within these groups, I'm part of the research group on advanced environmental applications of advanced oxidation processes, which is led by Professor Camila Costa de Amorim. And that's actually where I started I had the opportunity to work with contaminants of emerging concern. So by now, you're probably wondering, coming from such a tropical place with mango trees, why did I choose Minnesota? We now have like 11 Fahrenheit there. So my story with Minnesota actually goes back 10 years ago. So when I was an undergraduate student in biological sciences, I applied to this scholarship program by the Brazilian government, national government, that would pay, give scholarships to undergraduate students to come to the US for a year during undergraduate studies. And I applied, but I didn't know which university in the US within the program was going to pick me. And it turns out the University of Minnesota was interested in my profile. So I was an undergraduate student 10 years ago at the University of Minnesota. And at the time, I didn't know where Minnesota was. So I just looked at the map and I saw, it's a cold place near Canada. And I was in my early 20s, I would like to go somewhere warm, like California would not be bad. My mom is an academic, and I think she was just afraid I was going to turn the opportunity down. She started talking to her colleagues. She's also a biologist. And she found out that one of her colleagues actually had done his entire PhD here at the University of Minnesota under the supervision of Professor Jim Perry. So even before I came to Minnesota back then, I contacted Professor Jim Perry, and he was very open. He's very interested in internationalization and study abroad. And he told me about the classes he gives on water quality, and I made sure I would register to all of them. And it turns out I now work with water and water quality. And it was actually in one of Jim Perry's classes that I was introduced to the concept of contaminants of emerging concern. So just like Steve Jobs says, you can only link the dots looking backwards. So I think it was just meant to be the fact that I came to Minnesota 10 years ago. And then fast forward to 2021. It's the pandemics. We are all bored teaching online. And I ask a question, everyone has their cameras off, no one answers. And I'm like, okay, what I'm gonna do here? And then I see the Fulbright call. And it's a light at the end of the tunnel. Something just tells me go back to Minnesota. And so I wrote to Professor Jim Perry, and he said, well, I'll be in my sabbatical that year, but what kind of project would you like to propose? And then I knew, as I had been working with contaminants of emerging concerns since my master's, I knew PFAS was on the spotlight a year ago. So and still are. And so I told him, well, I would like to propose something with PFAS because there are not many studies in Brazil. So he put me in contact with Professor Matt Simsick from the School of Public Health in the Environmental Sciences Division, and also with Gerald T. Enkley from the EPA, who is an ecotoxicologist. And we proposed a project related to the occurrence of PFAS in an urban lake in my town. It's actually an urban lake that it's really close to the soccer stadium and to our campus. And so the project was approved, and now I'm here. So that's the story about my story with Minnesota. And now going back to contaminants of emerging concern. So as I told you, the first time I actually heard about it was in one of Jim Perry's classes back in 2013. And then during my PhD, I knew I wanted to work with CECs because as a biologist in the environmental engineering field, I knew it was a theme that I could link biology with engineering very closely and very tightly. And that would be easier for me in the future if I wanted to continue in academy or become a professor or a researcher in the area. So during my PhD, I worked with CSE occurrence control in wastewater and water, their fate in the environment, and also ecological risks associated to these CECs. And it was actually right by the end of my PhD that the research group approved a project funded by the Bill and Melinda Gates Foundation, which goal was to control antimicrobial resistance in wastewater by using advanced oxidation processes. So this funding was very important to us because it is a funding coming from an international association. And at this time, antibiotic resistant bacteria and genes had already been recognized as contaminants of emerging concern. So I'm talking about these contaminants of emerging concern. Most of you probably already know what they are, but some of you might not. So these contaminants are chemical compounds and other substances that were recently discovered in the environment. They've been there for years because we've been using them for years, but it was only after the evolution of analytical chemistry techniques that we were able to detect their occurrence in water, soil, and in sediments and the atmosphere. Although they're still not all included in routine of monitoring programs, and they might be included considering their ecotoxicity and effects on animal and human health. And so there are still no standards applied to most of them. In Europe, we already have some standards to some of them, and also in the US as well, but in developing countries, there are no standards whatsoever. And they are also named emerging contaminants, micropollutants, xenobiotics, so they have many different names that you could look for these pollutants in the literature. These contaminants usually come from industrial production like solvents, additives, flame retardants, lubricants, human consumption such as detergents, personal care products, and illicit drugs and pharmaceuticals, and also agriculture and livestock such as agrochemicals, antibiotics, and endocrine disruptors. You might be thinking, why did I add antibiotics under agriculture and livestock if we do take them as well when we get an infection? But it's because more than 80% of antibiotic usage worldwide is actually associated to agriculture and livestock production. And this excessive use of antibiotics actually leads to the selection and spread of antibiotic resistant genes and bacteria in the environment. And so these contaminants actually classify as these other substances to which the concept of chemical contaminants of emerging concern also comprehend. So ARBs and ARGs are also contaminants of emerging concern. Once in the environment, in the soil, air, water, or sediments, these contaminants go through different processes such as absorption, sorption, photolysis, and biodegradation, which contributes to their reduced concentration in these matrices. So these compounds, which might also be found as micropollutants, and this name comes from the fact that they occur from concentrations from nanograms to micrograms per liter. But even being present in these low concentrations in the environment, they've been associated to many different effects, such as feminilization and behavior changes in fish, neurological, reproductive, and immunological disruption in animals, selection of antibiotic-resistant bacteria and genes, and also obesity, heart disease, and diabetes type 2 in humans, not to mention all the effects they have in aquatic, terrestrial, and terrestrial biota in much lower concentrations, usually chronic effects that will only be visible at the but after completing the entire life cycle of this species. We also care about these compounds because they become ubiquitous in the environment. This study analyzed the occurrence of active pharmaceutical ingredients in 258 of the world's river, which are actually influenced the lives of more than 471 million people, and they found by sampling more than 1,000 locations and analyzing 61 actively pharmaceutical compounds. They detected these compounds in all of them. And carbamazepine, metformin, and caffeine were the most detected. They were present in more than 50% of sites. What is alarming is the fact that their concentrations of at least one of these compounds at 25.7% of the sampling sites was higher than the concentration which is considered safe for aquatic organisms, or higher than the level of concern for antimicrobial resistance. And one other conclusion they got to was that there was highest cumulative API concentrations in sub-Saharan Africa, South Asia, and South America. And you might be wondering why. So, well, when we think about the urban water cycle, these compounds usually come from households, industries, and effluents from manure. And when there is no wastewater treatment plant, they are directly discharged into the environment. And this is the reality for 50% of occasions in these regions. So sub-Saharan Africa, South America, and Asia. However, this does not mean that when there is a wastewater treatment plant, we are safe from the occurrence of these compounds in the environment, because municipal wastewater treatment plants are not designed to remove these contaminants. They're designed to remove organic matter and nutrients, but not removing contaminants of emerging concern. So they also occur in surface water, soil, and sediments in developed regions, but actually in a lower concentration. And then this wastewater treatment plants, they actually lead to only partial removal of contaminants of emerging concern via biodegradation. And depending on the technology, this removal might be different. So this heat map shows removal of different contaminants of emerging concern from 100% in green to 0% in red. When it comes to the activated sludge process, you can see that some of them are fairly well removed, but some of them are on the rate between 0 and 20%. For alternative technologies such as polishing ponds, we can see that there is not much data about it. And this is alarming when considering the reality of Latin American and Caribbean countries, because they rely mostly on these alternative technologies in municipal wastewater treatment plants. So stabilization ponds, they account for 38% of municipal wastewater treatment plants in Latin America and the Caribbean. And in Brazil, the anaerobic upsludge anaerobic upflow anaerobic sludge blanket systems actually account for 37% of our municipal wastewater treatment plants. Just a short comment, Brazil can still only treat 49% of the wastewater. So 51% is directly discharged onto the environment, and the other 49%, which are being treated, are treated mostly by alternative treatment processes such as WASP systems and polishing ponds, which are much less efficient in removing these contaminants of emerging concern, or we don't know what their efficiency is. So there is lack of data for these treatment systems applied in Latin America and in the Caribbean, considering their removal of contaminants of emerging concern. When it comes to antibiotics, this is even more critical. So we can see for most antibiotics here in this graph, there is no data on their removal by alternative technologies applied in wastewater treatment plants. And as a result, we actually have higher concentrations of antibiotic-resistant bacteria and antibiotic-resistant genes in the output of wastewater treatment plants compared to the untreated wastewater that gets to these sites. Once antibiotics are not biodegradable, and in the biological reactors, there is also there's actually a selective environment that will promote their reproduction and actually duplication, in a higher rate when compared to non-resistant organisms, and also higher transfer of antibiotic resistant genes between bacteria. So many research groups worldwide are working on the investigation of advanced technologies applied as post-treatment of municipal wastewater treatment plant effluent for the removal of contaminants of emerging concern, thus limiting or controlling their discharge to the environment, and controlling their effects to aquatic fauna, and also antimicrobial resistance spread. This is actually the whole figure made by Danga 2020, and here is the activated sludge process that I showed you earlier. But here we have advanced oxidation processes, which are the processes we work with in the research group. And you can see that they remove mostly, for most compounds, more than 80% of contaminants of emerging concern present in effluent. So they've been considered as promising alternatives, as they remove more than 80% of CECs present in the effluent. But most of this data, again, was acquired by applying this process to effluents from activated sludge systems, while in Latin America and the Caribbean, as according to the latest review we've published at Science of the Total Environment, they apply alternative technologies, and the effluent that comes from these alternative technologies, such as polishing ponds and anaerobic treatment systems, they have different physical chemical characterizations, such as higher conductivity, turbidity, organic content, inorganic content, and this influences the effectiveness of advanced oxidation processes, as salts and inorganic carbon. They actually compete with these contaminants for the oxidants that are formed in these processes. So as I'm talking about advanced oxidation processes, what are these processes? So advanced oxidation processes are advanced treatment processes that actually aim at the production of hydroxyl radical, which is this chemical species that is represented here. And this chemical species, it is it has a high redox potential of 2.8, and therefore it's very reactive and non-selective. So it will react with antibiotics, antidepressants, agrochemicals, and all these chemical compounds. And in ideal conditions, it will degrade them into smaller carbon chain organic compounds in ideal conditions, leading to CO2 and water, which are non-toxic and biodegradable. Recent studies have actually also worked with sulfate radicals, so replacing the predominant oxidant that is formed in the system. And this one has some advantages because it's a little less reactive, so it was be present in the system for a longer time, leading to sometimes, depending on matrix composition, leading to higher degradation of contaminants of emerging concern. So when compared to other advanced technologies, which actually lead only to phase transfer or concentration, such as adsorption or membrane filtration, and are energy intensive, advanced oxidation process, they promote the oxidation of organic compounds, toxic organic compounds, to smaller carbon chain compounds, leading to complete mineralization or biodegradable compounds. Simultaneous disinfection and degradation of contaminants of emerging concern may occur because these radicals, they also attack organic compounds present in bacteria membrane and inside bacteria cells. So they lead to, they induce damage to bacteria cell, and after accumulated damage, the cell actually dies. So there's damage of ARB cell constituents leading to death, and also ARG degradation. So they are also capable of reacting with purines, and they also enable the use of sunlight. And when it comes to countries such as Brazil and other tropical developing countries, it is a very important option to be able to use sunlight as an energy source for these compounds. So the process that we are currently more intensively investigating in the research group is called solar photophantom. In this process, iron salts react with hydrogen peroxide, forming hydroxyl radical. In this system, there is also the recycling of the iron 3 that is formed during this reaction, along with the hydroxyl radical with hydrogen, which reacts with H2O2 to form, to recycle iron 2. So iron 2 can go back to the system and then start the formation of hydroxyl radical all over again. The second reaction, or the cycling of iron 2, it actually occurs also in the dark, but it's faster under sunlight. And also under sunlight, there are some iron species present in the system that can absorb light within the solar spectrum. And this leads to an additional path for the formation of hydroxyl radical, thus making the process more efficient under light than in the dark. And with the need for a lower concentration of iron and hydrogen peroxide to reach the same efficiency of CEC degradation. Alternatively, we can also do the photophantom-like process, in which we replace hydrogen peroxide by persulfate, and then instead of forming hydroxyl radicals, we will form sulfate radicals, which, as I told you, is less reactive, so it remains for more time in the system, thus leading to more damage to bacteria cell and also higher degradation of contaminants of emerging concern. Also because they are less reactive also towards those competitors like inorganic and compounds and some salts, which are present in wastewater and natural surface water. So there is less competition between target contaminants of emerging concern and this matrix components for the radical formed in the system, usually leading to higher removal of these CECs. These processes, they have been proved effective for CEC removal and disinfection, mainly from activated sludge effluent, but there are still some challenges. So according to this recent review by Risu 2022, he's a researcher from Italy who is very active in the AOP world. There is still possible formation of toxic transformation products. So as hydroxyl radical and sulfate radical react with these contaminants of emerging concern, forming contaminants with lower carbon chains, so smaller carbon chains. Some of the smaller carbon chain compounds formed, they might be more toxic than the original CEC. So it's really important to follow toxicity to aquatic organisms throughout the treatment so that you know when to stop your reaction. Also, ARB and ARG removal have only been investigated for effluent from activated sludge and mainly at laboratory scale. And there is also limited pH for operation of this process because, as you probably know, iron at natural pH, which is pH 7, which is the pH of every river, lake water, most river and lake water, depending on the soil characteristics, it's actually in the solid state. But for this reaction to occur, it must be dissolved in the system. So usually, what we have to do is decrease the wastewater pH to acidic pH and then do the treatment and then neutralize it again. But there are some alternatives that we've been testing so that we don't have to add these two steps, acidification and neutralization, before and after respectively treatment. So the research group on environmental applications of advanced oxidation processes, which is the research group I'm part of at the Federal University of Minas Gerais, has the goal of applying AOPs for the removal of contaminants, conventional contaminants, so organic matter and nutrients. So as you probably know, aerobic treatment systems and alternative treatment systems applied in Latin America and the Caribbean, they don't lead to high removal rates of organic matter. So there's still a high content of organic matter remaining in the final effluent of these processes. So these radicals are also able to remove, we investigate if these radicals are also able to remove this remaining organic content. Also, and most importantly, contaminants of emerging concern, disinfection, ARB and ARG, and also toxicity. So we apply our solar photophantom aiming to remove or to reduce all of these effects or contaminants. And we do that not only to municipal wastewater treatment plant effluent, but also to effluents from industrial processes such as textile wastewater, pharmaceutical wastewater, and hospital wastewater, which have actually a higher concentration of antibiotics, antibiotic-resistant bacteria, and genes. So we want to reduce the load of ARBs and ARGs in this effluent before they are discharged onto the sewage system and go to the wastewater treatment plant. So we apply these AOPs, either aiming at discharge or reuse of this wastewater. And we usually do that by starting treatment at laboratory scale. So we have this solar chamber in our lab, which is a chamber that has a lamp. It's a xenon lamp that actually simulates sunlight. And then we get real wastewater at the municipal wastewater treatment plant. We take it to the lab and we do the treatment inside the solar chamber until we know what are the optimal concentrations of iron and hydrogen peroxide to use in the process that will lead to the highest removal of contaminants of emerging concern, bacteria, ARB, and ARG without increasing toxicity. Once we know what the optimum condition is, we actually scale up treatment to pilot plant reactors, semi-pilot plant reactors we have on the rooftop of the engineering school building. so that they are exposed to natural sunlight. So it's less control. It's not as controlled because, you know, the sun can be interrupted by clouds, and each day we're going to have a different sun radiation intensity. So we optimize first at lab scale, and then we go to the semi-pilot scale. This is one of the reactors we have. It's the compound parabolic collector. You can see that it has an angle which is equivalent to the latitude of our town, 20 degrees, and it has an aluminum surface in the back of this tube. So the effluent actually circulates in these tubes, and as sun radiations, sun rays, they reach the aluminum reflective surface and they accumulate in this tube. So it accumulates 100% of sun rays that reach the reactor, either directly at 90 degrees or diffusely at any angle different than 90 degrees, because they will reach the reflective surface and accumulate in the reaction tube. Temperature during this treatment can increase up to 60 degrees. So we start at 20 degrees and then we do like 2-3 hours of treatment, and temperature will have increased due to the concentration of light and heat. And what we do usually, so we start at lab scale, scale up to semi-pilot scale. To overcome the pH problem, we work with the intermittent iron addition strategy. So we apply iron throughout treatment at different times so that we can guarantee there is dissolved iron throughout the whole treatment, available to react with hydrogen peroxide or persulfate throughout the entire treatment without the need of decreasing the pH. And we spike our matrices with 100 micrograms of each of the target species we're going to work with. So instead of analyzing the removal of contaminants in their existing concentration, which is analytically very hard and expensive to do because you have to prepare your sample in many stages afterwards, we spike the matrix so that we can follow more easily and cheaper this initial concentration, the degradation of these compounds, starting at 100 micrograms per liter. So far in activated sludge effluent, we have proved that these processes, they are effective to remove more than 60% of total CECs in semi-pilot scale, so under natural sunlight using real wastewater. The treatment with persulfate, using persulfate as an oxidant source, a peroxide source, was cheaper and faster because it led to a lower reactor area. So as it's less reactive with matrix components, it was more reactive towards contaminants of emerging concern, thus requiring lower reactor area and being a cheaper treatment. And it did not generate any toxicity to luminescent bacteria, Allevibrio fischeri, after treatment. Considering anaerobic effluents from anaerobic systems, the traditional solar photophantone, which is the one that used hydrogen peroxide, was able to remove more than 60% of the contaminants of emerging concern, yet on a longer treatment time compared to activated sludge effluent, because these matrices, as I told you, have higher natural content of organic matter, as anaerobic systems are not as effective as aerobic systems to remove organic content. There was no toxicity to luminescent bacteria, Allevibrio fischeri, after treatment. And removal of phytotoxicity, which is the toxicity to plants, was also observed after treatment. This is really important when it comes to possible reuse of this effluent in agriculture for irrigation. Although it failed to remove toxicity to green algae, these results have not yet been published, but the student is working on the publication. But now what I want to call attention to is our latest results with antibiotic resistant bacteria and genes. So we, through the Bill and Melinda Gates Foundation funding we were able to produce some interesting results applying this process to effluents from activated sludge. So in this first study, we applied 30 milligrams per liter of iron and 50 milligrams per liter of hydrogen peroxide under sunlight. And this is the abundance of different phylum of bacteria in the untreated municipal wastewater effluent. The effluent treated by the traditional solar photophantom, and then only iron, and then only hydrogen peroxide and sunlight, which are the controls for this process. As you can see, there was a big change in the community in the abundance of different phyla. So what we observed was 74 to 86% removal of these groups. However, there was a fourfold increase in proteobacteria, which is the one in pink. And this is a concern because it is a group that accomplishes some of the whole priority pathogens when it comes to antimicrobial resistance. According to the literature, there was also proteobacteria selection after ozonation. So it seems like proteobacteria like oxidative environments. So when we looked at that a bit closely, we saw that groups such as Pseudomonas and Stenotrophomonas were the ones that increased the most after our treatment. And there was highest increase. So there was highest increase of gamma proteobacteria, to which those two groups belong, and especially Pseudomonas and Streptomonas, which are groups that have many known and hazardous resistant bacteria. When we looked at the whole genome sequencing of treated samples, we actually observed an increase of genes which are related to the scavenging of hydrogen peroxide in cells. So that was the mechanism they used to survive in this hostile environment in the presence of hydrogen peroxide, and that may be why they were selected during our treatment. Fortunately, this Pseudomonas aeruginosa, which is one of the whole priority organisms that carries it's a multi-resistance bacteria that promotes pneumonia, and it's really hard to treat actually. It was present in the untreated sample, but it was totally removed in the treated sample. So we are not able to find that in the treated sample. And Stenotrophomonas maltophilia, which is an emerging pathogen responsible for high morbidity and mortality, was also totally removed by our treatment. And so were a few other priority organisms. We also tried the photophantom-like process, not only using persulfate in place of hydrogen peroxide, but also using the mix of both oxidants. This is actually the first work in the literature that used the mix of these two oxidants. Interestingly, this time we did not see the increase of Pseudomonas. We had actually 88% removal of proteobacteria and 99% removal of bacteroidetes, but we actually had the selection of Firmicutes, which is a group formed mainly by gram-positive bacteria that has a thicker layer, so it might be more resistant to this treatment under the presence of persulfate, because as it remains for more time in the system, and these organisms have a thicker membrane layer, they were selected during this treatment. Interestingly, also, there was major DNA damage in the process using both oxidants simultaneously, so we were not able to assess its effect on ARGs through whole genome sequencing. And so this is a heat map representing genes which confer resistance to different antibiotic classes. And this column represents their abundance in municipal wastewater treatment plant effluent. This column represents their abundance in the sample after treatment with persulfate. This column with persulfate, but without sunlight. And this column in the presence of both oxidants, but without sunlight, only iron, because as I told you, in the presence of sunlight and both oxidants, DNA was damaged in a way that we could not perform whole genome sequencing. So for ARG removal, the municipal wastewater treatment plant effluent without advanced oxidation treatment, it had 74% of genes which confer resistance to sulfonamides, 10% macrolides, 9% tetracyclines, and 6% aminoglycosides. The removal by solar photophotone using hydrogen peroxide led to 55% removal of ARGs related to sulfonamides, 61% removal of ARGs related to macrolides, and more than 99% removal of ARGs related to beta-lactamic antibiotics, which are the most used in hospitals in South America. For treatment with persulfate as an oxidant, there was more than 50% of ARGs associated to broad spectrum antibiotics, and there was severe fragmentation. I'm sorry, there's an F missing here of DNA in the presence of both oxidants. So we were not able to do the WGS, and this is promising because it might be able to eliminate a higher content of ARGs. And considering the spread of antimicrobial resistance from wastewater discharge, it's also very important to remove plasmids, which are free in the wastewater or present inside the bacteria cell, but not connected to the DNA, and carry these resistance genes. Why? Because they may be exchanged from one resistant bacteria to a non-resistant bacteria, thus increasing the number of resistant bacteria in a sample. So in this other work, what we did was investigate if this hydroxyl radical was also able to damage and degrade this plasmid. So we used qPCR and bacteria transformation as methods. So we got two bacteria which had marked plasmids, so PSB1A2 and PSB1K3. And we applied our treatment. So these are the plasmids before treatment. And then we applied our treatment to see if these oxidants were able to degrade these plasmids. And what we found was a high removal of or degradation of plasmids within 30 minutes and 1 hour of treatment. But maybe these plasmids were broken, but they are still active. So what we did was we got a sample from this water that was treated with the non-bacteria And we incubated in plates containing the antibiotics in case this bacteria grew was because they were still resistant. So the breakage of the plasmid was not enough to inactivate the plasmids. But what we found was that there was no growth after our treatment. So the treatment was efficient to break and to inactivate this resistant conferring plasmids. So some final remarks. I did speak more than I should have, but considering basic sanitation, there is still a lot to improve in Brazil before CEC standards can be set. CEC control by AOPs in Brazil, we've been advancing in the past years in our research group and also other research groups with which we collaborate in the country. But there are still major gaps considering the removal and transformation of CECs, including ARBs and ARGs, especially in effluents from anaerobic treatment systems. And this leads to many different opportunities of collaboration between U of M and UFMG to bridge this literature gap. So I'm open to collaboration. Let me know if you have any questions. You can send me an email. So this is my UMN email and then my email from my institution in Brazil. I would like to thank the BBE program and then Fulbright, the University of Minnesota, my own institution, and the research group for having the opportunity to be here today. And that was all I had to say. Thank you.
Yeah, thank you, Maria. I think we have 2 minutes left. I don't know if
Yeah.
stay here for questions or you want to just.
Yeah, well, I have to go to where the event is, and it actually starts at 5:30. And Brazilians have this fame of being late to everything, and that I don't want to cope with this. So
Yeah, sure,
I
sure.
think I will say
Yeah.
my goodbyes. And I mean, as I told you, I'm really opening for collaborations, and I really want to, to give you the opportunity to go to Brazil too, as I'm here at the University of Minnesota. I was very well hosted both times I came. So I'll be glad to answer you and to propose new projects also in these and other areas.
Yeah. So I think we do have a lot of shared interests, between you and our departments. Actually, a lot of the groups in a department who study the bioremediations, and especially some are here and or they're leaving. But so I think we share the interests a lot. Yeah. So yeah, thank you.
Thank you.
It's a fantastic.
Thank you for the opportunity. Thank you, everyone.
Yep.
Bye bye.